Thank you all for the invitation here this year. So, slide that you see on the screen now, this title slide is not my slide. It's the title slide from 1 year ago at the forum, and where the director of NIDCD Temtucci gave a talk, and that talk was entitled "An Introduction to FaceBase's Newest Partner, NIDCD and the Temporal Bone." This year, it's my job to recap our progress over the last year on this project we call EarBase, which is being added to FaceBase as a new consortium starting June the 1st. First, a bit of context why we're doing all this. So FaceBase, EarBase, in particular, EarBase is all about the temporal bone, and the temporal bone is a busy place. Anatomically, the organs of hearing and balance are embedded in the temporal bone. Also here are jugular vein and the internal carotid, which flank the middle ear space on both sides. Also passing through the middle ear is the facial nerve, or parts of the facial nerve. One of the branches supplies muscle expression, or innervation to muscles for facial expression, over the entire face. Another branch, the chorda tympani, passes directly through the middle ear space and innervates the tongue for taste sensation. So there's a lot going on here, not just auditory and balance. On the right is an image which I'll come back to later, but these are 10 micron sections through the temporal bone. And they're stained for hematoxylin and eosin, and we have thousands of these images that contain the cochlea. You can see in cross section here, the middle ear space where the 3 middle ear bones are located. This is the external ear canal, and then there's an internal auditory canal, which is the passageway from the 8th cranial nerve to the brain. So it's images like this one that are currently at least the primary data types in EarBase. I would like to also provide some additional context here. The NIDCD Temporal Bone Initiative is an effort to facilitate research in human hearing and balance disorders by integrating 3 different resources. We have a nationwide donor program, and that program manages recoveries of temporal bone, also brain and DNA from consented donors. There's a network of research laboratories. There's 5 labs in that network right now. They conduct basic and applied research in hearing and balance disorders. And then there's EarBase, which is the new database that's intended to serve as the hub for consolidation of all the data being generated, and also a place for data sharing and tissue sharing. So a bit more context, a little history. The Temporal Bone Initiative evolved over several decades. Support from NIDCD, several foundations, and dedicated teams of clinicians, researchers, and technicians who, over several decades, collected temporal bones and process them and preserve them into what will be the resource that we're putting together now. So in terms of databases, along the way, there was a database that was established by one of the research labs in the network, Mass Eye and Ear. And that database primarily contained their holdings, as well as some fragmented pieces of collections distributed across the other sites in the United States. And in the transition to EarBase, as we were consolidating all of the data, we think now that there are data for about 9,000 human cases distributed across the country, with new cases being added every year. So the pros of that, the good news on that collection is that most of those patients have a positive history of otologic disease. So the value of the data sets is quite high. The con is that Many of these collections are distributed across the United States and haven't yet been consolidated. So a little bit more about the data. What are they? Well, they're patient data, demographics, clinical histories, especially focused on diagnosis and treatment of hearing disorders. There's histopathological data, primarily on the temporal bone, tissue assays, digitized images, analyses of those digitized images, and also DNA and brain are available on a subset of cases. So more about that later. A few stats. So we think there are 9,000 cases plus the new arrivals. About 2/3 of the cases that are in the records are in the hands of the network labs. So that's about 6,000 cases. That translates into about 500,000 H&E sections for those individuals, and about 3 million of those are unused. 1 in 10 sections through the temporal bone are stained for H&E. The other 9 are reserved for whatever purpose. So that becomes significant. So the point is that even if we only had access to half of these data, the value of this data set collection is really enormous. But as I mentioned before, this is exciting, but the data have never been assembled into one place. So that's where FaceBase and the EarBase Consortium enters the picture. So the release on June 1st will be the first time that these data have been consolidated into one single spot and made accessible to the research community. The June 1st launch will be a little bit more of a soft launch. Some elements will be complete. Some of the uploading of files is still ongoing. It's been an onerous process, and I don't know. I think of it a bit like visiting Disneyland when they have some new attractions that are under construction. There's still a lot to do at Disneyland, and visitors will have a lot to do on EarBase, but there's a lot to look forward to also. So that's where we're at with EarBase. We have a good start, but much of what's coming is in the future. So in the last year, we've been doing 3 main things with the FaceBase team. We've been building a data model that fits in with the FaceBase environment and a user interface that accommodates the unique aspects of the temporal bone data. And I'll just pause to give a shout out to Alejandro and Carl and Robert and others on the FaceBase team for all of the pain and suffering that we've inflicted upon them to put this together. They've had to bend and help out a lot to put this thing together. So data model, the user interface, and then the data uploads are in progress and ongoing. So I'd like to take a bit of time to share with you an overview of the user interface and the data model. It's built on the FaceBase platform, so it has some familiar aspects. Project level, there are 5 projects. You probably can't see that very well, but the 5 network labs that are initially contributing are Johns Hopkins, Mass Eye and Ear, UCLA, the University of Minnesota, and Stanford. And each of those are contributing primarily human data sets for now, but they also have animal data sets of various kinds. Another level that's been added to the user interface is what we consider the case level. And a case is equal to a single donor or a single patient. So what we have for these cases are demographic information, diagnostic, histopathologic information, and also information about any treatments that have been performed and other clinical history that's available. A 4th level is what we consider to be the specimen or biospecimen level. Primarily right now, that's the temporal bone. So this level contains details about how the tissue was acquired, how it was processed, how it was stored, records of tissue usage, what assays have been done so far. And also part of that is the capability to track tissue usage to support collaborations and sharing. As I said, a great number of tissue sections are still available for other types of assays. Then finally, the files that are part of the data set include, for the case side, we have audiograms and other tests of hearing and balance for individuals. Clinical imaging, when we can get it, like CT scans are pretty common in hearing and balance assessment, other tests, as well as the images and other data. So all that's there and downloadable. A bit more about the temporal bone and temporal bone sections. So the H&Es are high resolution scans, and each ear, each temporal bone contains about 40 to 50 H&E sections. And these are viewable that EarBase launches as thumbnails, and also in a full resolution viewer that the team is building in, so that someone can look through the H&Es and zoom in and see what's there. And in zooming in, one can zoom into the level of individual cells. So analyses and viewing is possible down at the single cell level. And one can also put together the stacks of these images, 40 to 50 per ear, and create 3D constructions that are also analyzable down to the single cell level. So if 1 in 10 sections are stained for H&E, the remainder can be used for other purposes. These are some pretty images from some of the network labs where immunohistochemistry for various targets was applied to look at some of the fine structure of axons, cells, stereocilia, etc. So these features are examples of what can be done with the additional tissue sections. In addition to that, our volumetric imaging of postmortem tissue samples, the temporal bone, dissected out, and typically CT, micro CT, and some other imaging tools are being applied and optimized for use in temporal bone. The temporal bone is the organs of hearing and balance. It's a really small part, and so it requires special imaging techniques to get down there. But you see on the bottom two panels here is examples of synchrotron imaging from the Stanford lab and a 3D fly-through of the cochlea that they were able to create there. So the point is, regardless of the type of imaging, it permits evaluations of structural integrity of the temporal bone prior to histology, or in some cases in lieu of histology. Back to the data model, I want to pause to mention something about data curation. So we think that one of the most important features of the resource that we're building is achieved by very careful curation of the data at the case level, so for each individual donor or patient, and also at the specimen level. Because many of the records that we have right now are incomplete, some going back to the 1950s or older, a great deal of effort will be ongoing to fill out and complete the curation at a level that we would like to see. So I know you won't be able to see what's in this slide very well, but this is a page from a single patient showing a bunch of demographic and diagnostic information. And this works here. Yeah, there we go. So here's an excerpt from that page where there's diagnostic information for that patient. There is treatment information for that patient. And then there is post-mortem histological evaluation of the temporal bones from that patient. So at least at the case level, this is the level of detail at which the data are being curated, and it's all fully searchable, completely searchable, and it can be used then to narrow down selection of particular cases for study. So what about DNA and other omics? Well, it may be useful context to know that about 50 to 60% of children with hearing loss are due to a genetic mutation of some kind. About 80% of those are non-syndromic. The other 20% are syndromic. And at the current time, we know of about 120 genes, typically single gene mutations, that are responsible for various forms of hearing loss. You may have known recently, last week, the FDA announced a gene therapy approval for one of those genes, otoferlin. So it's a major focus of NIDCD and the work of its researchers. On EarBase, what we will have in the beginning are indexes to DNA that have been collected on patients that have also donated their temporal bones. Currently, the DNA is being analyzed by labs, typically stored by individual laboratories, may be uploaded to a repository. We'll be developing ways to represent the DNA on EarBase over time. As you heard yesterday, we already have a go-to transcriptomics database supported by NIDCD, and that's GEAR at Maryland. So I assume that will continue to go on, and the proteomics data we comment into EarBase. All right, and now a couple of comments about data sharing and reuse, since that's a major theme this year. So because EarBase is being established on FaceBase, the tools for data sharing and reuse are already baked into the platform. So our responsibility is to make sure that we're using standard formats for the images, and we are curating rich data sets from all available data, right, and establishing this tissue sharing mechanism so that the donated tissues can be used in the maximal way. So what does this kind of thing set up? Well, I'm going to give you a few examples. So the first is this. Let's say a typical EarBase user goes in, selects a certain clinical diagnosis, maybe severe hearing loss, and then proceeds to select some images and conduct analyses on the cochlea that focus on missing hair cells. That's a typical use case, and we expect to see a lot of that kind of thing. Okay, the next day, another user enters EarBase. They select the same patients because by selecting the same diagnosis, they analyze the cochlea, but they also optimize their image selection to analyze the auditory nerve. They do that, then they realize that the H&E stains can only get them so far with the auditory nerve. So they then reach out to the contributing laboratories for tissue sections that they conduct immunohistochemical assays on for certain targets. The next day, right, another user enters. They select the same diagnosis, but they also add a secondary diagnosis. Let's say it's type 2 diabetes, right? They conduct assays of the hair cells and the auditory nerve, histochemical assays, but then they decide that they would like to screen for novel genetic variants in the patients that have both of these conditions. So that's a third use case on the very same tissue sections from the same donors. And then one more. We know that EarBase will contain data from patients with disorders that affect both craniofacial and auditory or balance structures. Examples are Treacher-Collins or Crouzon syndromes. And so by exploring in parallel data sets on EarBase and FaceBase, that process will generate some new discoveries. And just to illustrate the potential of that, I would like to close my talk with a story. And it involves this gentleman here, Andy Groves. Now, Andy is the new head of developmental biology at WashU, recently moved from Baylor in the fall. Some of you may know him. But in our field, Andy has conducted research on inner ear development and regeneration for over 25 years. He's a legend in our field, but doesn't like to be referred to in that way. He's one of the nicest guys you'll ever meet. But I happened to be with Andy a few weeks ago, and I noticed that he had become involved in a new line of research, craniofacial microsomia. I didn't know about that, but of course it caught my attention right away. So as many in this audience will probably know, variants in the FOXI3 gene can cause a range of structural anomalies, the hair, the face, the teeth, but also the ear. So my question was, Andy is primarily an inner ear researcher. How did he become involved in craniofacial microsomia? So I'm going to read to you what he told to me, and it's his story, not mine. So here we go. A postdoc in my lab cloned all 3 mouse FOXI genes, 1, 2, and 3. We saw that FOXI3 is expressed in the progenitors of all craniofacial placos and in the pharyngeal ectoderm and endoderm. We made a FOXI3 mouse knockout. The mutants had no inner ear, no middle ear, no ossicles, no tympanic ring, no external ear, a severely truncated jaw. We were interested to know if there were any pathogenic human variants, so we used the Gene Matcher portal to get updates on any FOXI3 variants. We got a match from a lab in Switzerland that had 2 brothers with hemifacial microsomia and microtia, and a coding variant in FOXI3. It turned out that they were collaborating with my next door neighbor at Baylor, Jim Lupsky, who had found a couple of other families with the same condition. I then heard that Jim and the Swiss lab were working with the Chinese lab, who had about 20 more families with hemifacial microsomia and microtia. So both the lab in China and my lab had started making mouse versions of some of the pathogenic coding variants. We all got together and pooled our results and wrote the Nature Communications paper. Okay, these are great science stories like this. I love these kind of stories. And the reason I raise it in this context is that while we cannot claim this particular instance as a use case for the joint venture of EarBase and FaceBase, we know that there are overlaps between the 2 fields. Just may not know what they all are. And so the potential for generating these kinds of new findings that overlap between the 2 fields is quite great. So I'll close my talk by showing the acknowledgement slide, and thanks to everyone for taking the time today.
Okay, questions for Troy? Yes.
I don't want to mention that we actually have a very large study of microsomia from Washington University, from Carrie Hackett. It's a very large collection of data.
Well, that's good. So there's lots too.